So I'd like to talk a little bit about the cochlear structure, but in particular, some of its frequency dependence and some of the basic processing that you see as you work your way through the basic cochlear structure. So if you were to take a cochlea, which is sort of this sort of round structure that we see that's very important for hearing, what you end up seeing if you roll it all the way out is this interesting structure where you have sort of the stapes that pushes a pressure wave in. You have a pressure wave fluid going all the way through, comes back on the other side, and then ends up pushing out through the window. Okay, so that works out very well. And what you notice, a lot of the key things are looking at the basic membrane, particularly asking the question, what happens in terms of the membrane here, that sort of core basilar membrane at the bottom. And what you notice that all of these tend to be different sizes. So let's take a look at this a little bit more schematically. One can look at all the biological pieces of this, but there's an interesting question. And what you notice is that there's two sides to it, an upper side and a lower side, of which the fluid's going to maybe go forward into this and then approximately forward, pushing back on the other side. These are relatively uniform stimulations in terms of fluid. If you think about it, the fluid is incompressible, so it's going to pretty much be very similar on both sides. And you have one sort of membrane that kind of gives me a relatively uniform behavior all the way through. This works well. So here's the question. Then you look at the structure and you say, well, okay, now I really care about what's in the basilar membrane. And what it turns out is this has its own sort of mass and structure to it. That is has sort of spring-like structures on both sides. And so then there's pressure that comes through here. It's somewhat uniform, but then pushes against all of these elements. There's some that gets pushed on the back side of this. And that's an interesting question. Now, if you look at this, take this mechanical model of just this part of the basilar membrane, what you notice is that it's like I have two very strong sort of anchored positions on both sides. And then I have these elements all the way in the middle, and they're different sizes. In fact, as I am close to the front of this, it's very close to being, very, very small, so very high, it has a much higher resonant frequency. In fact, the resonant frequency is relatively sort of a fourth order based on the size of the structure. And it's interesting how these get scaled all the way through. And they'll have different masses that are all relatively independent of each other, although they're all connected through the fluid, so there's some interdependence to the fluids. And so certainly local dependence and local dependence over an entire structure. Still, there's some dynamics there. But here's what's interesting. And you can see this both in terms of frequency responses by, cochleas of sort of, they basically just extracted cochlear structures and then that you stimulate, or through multiple models or through multiple elements. And one of the things you see in the classic sort of data is if I look at sort of the characteristic frequencies of this, and again, this is sort of, these are pictures from other people's data that working with here. But if you look at the original characteristic frequencies, it actually ranges over this sort of, 100 hertz to, over tens of kilohertz, depending on the cochlear structure, depending on the particular mammal that you're working with. And if you actually look at sort of its distance, so going all the way to the far end, which is all the way over here, to going at the very beginning, which is going to be at the higher frequency elements, you notice something very interesting, that overall this response goes basically exponential with position. A little bit of interesting issue at the low frequencies, maybe due to boundary conditions and other things not scaling, but it's relatively exponential, and this is sort of typical that you start to think about it. In fact, you look at the responses, it looks like a whole bunch of bandpass filters from that particular data set. There's many different cases. That's not shocking, with some resonance in there. It's not shocking if I was saying I have a whole bunch of resonators with springs attached. This effectively looks like an RLC circuit. No surprise we're getting these kinds of resonance things. But what is very useful is to know that you have a relatively exponential shift in that corner as a function of position. And so what I would want to often have as a zeroth order approach is to have something that is going to be a set of bandpass filters that are exponentially spaced. And this is important because physical systems do this fairly naturally. Trying to do this in, say, something that would be more of a DSP model or a digital model actually is quite a bit of complexity. But all acoustic kinds of questions that are related to human hearing are eventually going to come through this question one way or another.